Welcome all to the video learning on M phase. Following the completion of S phase and transition through G2, the cell undergoes the dramatic upheaval of M phase. M phase of the cell cycle consists of nuclear division, that is mitosis, followed by cytoplasmic division, that is cytokinesis. The central problem for a cell in M phase is to accurately separate and segregate its chromosomes so that each new daughter cell receives an identical copy of the genome. With minor variations, almost all eukaryotic cells solve this problem in a similar way. They assemble specialized cytoskeletal meshes that pull the duplicated chromosomes apart and split the cytoplasm into two equal halves. Entry into mitosis orchestrated by MCDK. Remarkable features of the cell cycle control system is that a single protein kinase, that is MCDK, brings about all of the diverse and complex cell rearrangements that occur in the early stages of mitosis. This happens only after the activation of MCDK, which is maintained inactive by V1 kinases that phosphorylates the two active sites on the enzyme throughout the interphase. Once the cell makes up to G2 phase, the trigger to enter mitosis is sensitized by the activation of MCDK protein. The so far inactive MCDK is activated by CDC25 phosphatase, which releases the inhibitory phosphates from the active site, after which the activating phosphate is bound to the protein by CDK activating kinase enzyme, restoring MCDK activity, which then binds cyclin to perform functions like induce the assembly of the mitotic spindle and ensure that each sister chromatid in a pair is attached to the opposite pole of the spindle. Triggers chromosome condensation, the large scale reorganization of the intertwined sister chromatids into compact rod like structures. Promotes the breakdown of the nuclear envelope and rearrangements of the actin cytoskeleton and the Golgi apparatus. Each of these processes is thought to be initiated when MCDK phosphorylates specific proteins involved in the process. MCDK does not act alone to phosphorylate key proteins involved in early mitosis. Two additional families of protein kinases, the polo-like kinases and the aura kinases, also make important contributions to the control of early mitotic events. In preparation for M phase, DNA binding proteins configure replicated chromosomes for segregation. When the chromosomes are duplicated in S phase, the two copies of each replicated chromosome remain tightly bound together as identical sister chromatids. The sister chromatids are held together by protein complexes called cohesins, which assemble along the length of each sister chromatids as the DNA is replicated. This cohesion between sister chromatids is crucial for proper chromosome segregation, and it is only broken late in mitosis to allow the sister chromatids to be pulled apart. When the cell is about to enter M phase, the replicated chromosomes condense, becoming visible as thread-like structures. A set of protein complexes called condensins help carry out this chromosome condensation. The M phase cyclic dependent kinases, that is MCDK, that initiates entry into M phase triggers the assembly of condensing complexes onto DNA by phosphorylating some of the condensing subunits. The accumulation of condensins on the DNA facilitates the progressive condensation of the chromosomes. Condensation makes the mitotic chromosomes more compact, reducing them to small physical packets that can be more easily segregated within the crowded confines of the dividing cell. The cohesions and condensins are structurally related and they work together to help configure the replicated chromosomes for mitosis. Cohesins tie two adjacent sister chromatids together. Condensins coil up single DNA molecules in the process of chromosome condensation. The cytoskeletons mediate M phase. After the condensation, two separate cytoskeletal structures are assembled in sequence to carry out the two mechanical processes that occur in M-phase nuclear division and cytokinesis. Both structures are rapidly disassembled after they have performed their tasks. To produce two genetically identical daughter cells, the eukaryotic cells has to perform the delicate job of separating the replicated chromosomes and allocating one copy of each chromosome to each daughter cell. In all eukaryotic cells, this task is carried out during mitosis by a complex cytoskeletal machine called the mitotic spindle. The spindle is composed of microtubules and the various proteins that interact with them, including microtubule dependent motor proteins. In animal cells and many unicellular eukaryotes, a different cytoskeletal structure is responsible for cytokinesis. It is called the contractile ring because it consists mainly of actin filaments and myosin filaments arranged in a ring around the equator of the cell. The contractile ring starts to assemble towards the end of mitosis just beneath the plasma membrane. As the ring contracts, it pulls the membrane inward, thereby dividing the cell into two. The figure shows the two transient cytoskeletal structures mediate M phase in animal cells. The mitotic spindle assembles first to separate the duplicated chromosomes. Then the contractile ring assembles to divide the cell into two. Whereas the mitotic spindle is based on microtubule and the contractile ring is based on actin and myosin. Centrosomes. Before M phase begins, two critical events must be completed. DNA must be fully replicated and in animal cells, the centrosomes must be duplicated. The centrosome is the principal microtubule organizing center in animal cells. It must be duplicated so it can help form the two poles of the mitotic spindle which will separate the duplicated chromosomes and distribute them to the two daughter cells. Furthermore, the centrosome must be duplicated if each daughter cell is to receive its own centrosome. Each centrosome consists of an amorphous matrix of proteins containing hundreds of gamma tubulin rings. These ring complexes serve as nucleation sites for the growth of microtubules that radiate out from the centrosome. In animal cells, the centrosome also contains a pair of centrioles, each made of a cylindrical array of short microtubules. During interphase of each animal cell cycle, the centrosome is duplicated and both copies remain together as a single complex on one side of the nucleus. As mitosis begins, the two centrosomes separate and each nucleates a radial array of microtubules called an aster. The two aster move to opposite sides of the nucleus to form the two poles of the mitotic spindle. When the nuclear envelope breaks down, the spindle captures the chromosomes, eventually separating them in mitosis. Centrosome duplication is triggered by the same CDKs that trigger DNA replication. As mitosis ends and the nuclear envelope reforms around the separated chromosomes, each daughter cell receives a centrosome in association with its chromosomes. The process of centrosome duplication and separation is known as the centrosome cycle. Kinetochores. Kinetochores attach sister chromatids to the spindle. The kinetochore is a network of protein complexes that assemble on centromere regions of chromatin and acts as the connection point between chromatids and the spindle microtubules that segregate them into daughter cells. Kinetochores must sense microtubule attachment and determine whether this attachment of sister chromatids is to the same or different spindle poles. The stable engagement of a kinetochore is regulated by the aura B kinase, the activity of which has been shown to reduce the affinity of several kinetochore proteins for microtubules. A single unattached or incorrectly attached kinetochore is sufficient to trigger the spindle assembly checkpoint and halt progress into anaphase, preventing cell division. Once all of the chromosomes in a cell preparing to divide are correctly bi-oriented and satisfy the spindle assembly checkpoint, chromosomes must be pulled apart into daughter cells. An essential function of the kinetochore is to couple chromosome movement to microtubule depolarization. Kinetochores are able to track depolymerizing microtubule ends and harness the energy released during microtubule depolymerization to move chromosomes to opposite spindle poles. Following the assembly of a bipolar microtubule array, the second major step in spindle formation is the attachment of the array to the sister chromatid pairs. Spindle microtubules become attached to each chromatid at its kinetochore, a giant multi-layered protein structure that is built at the centromeric region of the chromatid. In this picture, the picture A is a fluorescence micrograph of a metaphase chromosome stained with a DNA binding fluorescent dye and with human autoantibodies that react with specific kinetochore proteins. The two kinetochores, one associated with each sister chromatid, are stained red. B. A drawing of a metaphase chromosome showing its two sister chromatids attached to the plus ends of kinetochore microtubules. Each kinetochore forms a plaque on the surface of the centromere. C. Electron micrograph of an anaphase chromatid with microtubules attached to its kinetochore. Stages of M phase. Mitosis is a cell division phase during which the sister chromatids are separated and distributed, that is segregated, to a pair of identical daughter nuclei, each with its own copy of the genome. Mitosis is traditionally divided into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase, defined primarily on the basis of chromosome behavior as seen in a microscope. As mitosis is completed, the second major event of M phase, cytokinesis, divides the cell into two halves, each with an identical nucleus. Prophase. At prophase, the replicated chromosomes, each consisting of two closely associated sister chromatids, condense. Outside the nucleus, the mitotic spindle assembles between the two centrosomes which have replicated and moved apart. Prometaphase. Prometaphase starts abruptly with a breakdown of the nuclear envelope. Chromosomes can now attach to spindle microtubules via their kinetochores and undergo active movement. Metaphase. At metaphase, the chromosomes are aligned at the equator of the spindle, midway between the spindle poles. The kinetochore microtubules attach sister chromatids to opposite poles of the spindle. Anaphase. At anaphase, the sister chromatids synchronously separate to form two daughter chromosomes and each is pulled slowly towards the spindle pole it faces. The kinetochore microtubules get shorter and the spindle poles also move apart. Both processes contribute to chromosome segregation. Telophase. During telophase, the two sets of daughter chromosomes arrive at the pole of the spindle and decondense. A new nuclear envelope reassembles around each set, completing the formation of two nuclei and marking the end of mitosis. The division of the cytoplasm begins with contraction of the contractile ring. Cytokinesis. During cytokinesis, the cytoplasm is divided in two by a contractile ring of actin and myosin filaments which pinches the cell into two to create two daughters, each with one nucleus. Thank you all.